Welcome to this StudyNeuro lecture on the pathogenesis of ankylosing spondylitis. This is the foundation. Understanding how the disease develops helps explain its clinical features and why modern treatments target specific immune pathways rather than simply relieving symptoms. In this session, we will examine the current understanding of disease pathogenesis, beginning with the role of the human leukocyte antigen B27 gene and other genetic risk factors, before exploring the major theories that link these genes to disease development. We will also discuss the interleukin 23 and interleukin 17 immune pathway, explain how chronic inflammation drives both bone erosion and abnormal new bone formation, and connect these mechanisms to the biological therapies that have transformed patient care. If you enjoy evidence-based neuroscience and clinical medicine, join the StudyNeuro community on YouTube, Facebook, TikTok, Instagram, and X for more high-yield educational content. Let's get started. Ankylosing spondylitis is a chronic immune-mediated inflammatory disease that mainly affects the sacroiliac joints, the spine, and the entheses, which are the sites where tendons and ligaments attach to bone. Its biology is complex. The disease develops through an interaction between genetic susceptibility, abnormal immune responses, environmental influences, and changes in the way bone is broken down and rebuilt, eventually producing both persistent inflammation and abnormal new bone formation. As the disease progresses, inflammation damages tissue while also stimulating excessive bone growth, a process that can lead to progressive spinal stiffness and ultimately spinal ankylosis. Understanding these mechanisms has changed clinical practice because it has led to targeted biologic therapies that interrupt specific immune pathways rather than simply controlling symptoms, resulting in more effective treatment for many patients. The development of ankylosing spondylitis is not driven by a single event. It is a multi-step process. The disease begins in people with a genetic predisposition, particularly those carrying the human leukocyte antigen B27 gene, and then progresses through abnormal antigen presentation, changes in the gut microbiome, and activation of both the innate and adaptive immune systems, which together sustain chronic inflammation. As these immune pathways remain active, the interleukin 23 and interleukin 17 signaling pathway becomes a major driver of inflammation at the entheses. This persistent inflammatory response causes bone damage while also stimulating abnormal new bone formation, eventually leading to spinal ankylosis. These biological processes continue to interact throughout the course of the disease, making ankylosing spondylitis a dynamic and progressive disorder. The human leukocyte antigen system is a group of proteins that helps the immune system recognize which cells belong to the body and which may be harmful. It is central to immunity. These proteins are encoded within the major histocompatibility complex on chromosome 6, and one member of this family, human leukocyte antigen B27, has the strongest known genetic association with ankylosing spondylitis. Human leukocyte antigen B27 presents protein fragments from inside cells to cluster of differentiation 8-positive T lymphocytes, helping initiate adaptive immune responses while maintaining immune surveillance and distinguishing self from non-self. About 90 to 95% of patients with ankylosing spondylitis carry this gene, yet many healthy individuals also test positive, showing that human leukocyte antigen B27 increases susceptibility but does not cause the disease on its own. How does human leukocyte antigen B27 cause disease? Human leukocyte antigen B27 contributes to ankylosing spondylitis through several biological mechanisms that appear to work together rather than acting alone. Current evidence supports the arthrogenic peptide hypothesis, the misfolding hypothesis, and the cell surface homodimer hypothesis. Although each explains a different part of the disease process, no single mechanism explains everything. In genetically susceptible individuals, these pathways interact to trigger abnormal immune activation, sustain chronic inflammation, and promote the tissue damage that eventually leads to structural changes in the spine. Arthrogenic peptide hypothesis. The arthrogenic peptide hypothesis proposes that human leukocyte antigen B27 presents certain self or microbial peptides to cluster of differentiation 8 positive T lymphocytes, triggering an immune response that mistakenly targets the body's own tissues. This abnormal recognition activates autoreactive T cells, which then sustain inflammation instead of resolving it. This is one proposed mechanism. Although it does not explain every feature of ankylosing spondylitis, it provides an important model for understanding how genetic susceptibility may initiate chronic immune-mediated inflammation. The misfolding hypothesis focuses on what happens inside the endoplasmic reticulum, the part of the cell responsible for folding newly made proteins into their correct shape. Proper folding is essential. Unlike most human leukocyte antigen molecules, human leukocyte antigen B27 folds slowly, increasing the chance that abnormal proteins will accumulate and place the cell under stress. This cellular stress activates protective pathways such as autophagy and promotes greater production of interleukin 23, which further amplifies inflammatory signaling. Although researchers continue to debate the exact contribution of the unfolded protein response, substantial evidence suggests that human leukocyte antigen B27 misfolding plays an important role in initiating and sustaining the chronic inflammation seen in ankylosing spondylitis. Misfolded human leukocyte antigen B27 molecules can join together to form abnormal structures known as homodimers on the cell surface. These molecules are biologically active. They interact with immune receptors, such as killer cell immunoglobulin-like receptor 3 domain long cytoplasmic tail 2 and leukocyte immunoglobulin-like receptors, altering the behavior of several immune cells. These interactions stimulate natural killer cells, support the survival of T helper 17 cells, and increase the production of interleukin 17 and tumor necrosis factor alpha, two cytokines that play major roles in inflammation. As a result, immune activation persists at the entheses and throughout the axial skeleton, helping to sustain the chronic inflammatory process that characterizes ankylosing spondylitis. ARAP1 and ARAP2 are enzymes found inside the endoplasmic reticulum, where they prepare protein fragments before these fragments are placed onto HLA class I molecules for recognition by the immune system. This step is very important. When these enzymes do not work as they should, the protein fragments are processed differently, which changes how they are presented to immune cells, encourages misfolding of human leukocyte antigen B27, and can drive a stronger immune response that makes inflammation more likely. Research has shown that changes in the endoplasmic reticulum aminopeptidase 1 gene are the second strongest genetic risk factor after human leukocyte antigen B27, and their effect becomes greater when both genetic changes are present together, increasing the chance of developing ankylosing spondylitis through their combined action. This interaction is well established. The interleukin 23 and interleukin 17 pathway is the main inflammatory pathway in ankylosing spondylitis. It drives much of the immune response that leads to persistent inflammation and later structural damage. This pathway is central. The process begins when immune cells present an antigen, which stimulates the production of interleukin 23. This cytokine promotes the development of T helper 17 cells, and these cells release interleukin 17A and interleukin 17F, which then trigger the release of other inflammatory cytokines, including tumor necrosis factor alpha, interleukin 6, and interleukin 1 beta, creating a self-sustaining inflammatory response that continues over time. As inflammation persists, damage gradually develops in the joints and surrounding tissues, eventually leading to abnormal bone formation and reduced spinal mobility. This explains why drugs that block interleukin 17 are effective treatments for many patients with ankylosing spondylitis. Early treatment matters.
Ankylosing spondylitis develops through the combined actions of the innate and adaptive immune systems, which normally protect the body from infection and help regulate immune responses. Many immune cells take part. Dendritic cells, macrophages, natural killer cells, and innate lymphoid cells help start the inflammatory response, while different T cell populations continue and shape that response over time. The adaptive immune system then plays an important part through cluster of differentiation 4 positive T cells, cluster of differentiation 8 positive T cells, T helper 17 cells, and regulatory T cells, while B cells appear to have a smaller role. And together, these cells release inflammatory signals that keep inflammation active and promote ongoing tissue remodeling. This process drives disease progression. The gut-joint axis describes the close connection between the immune system in the intestine and inflammation in the spine and joints. This link is increasingly recognized. Many people with ankylosing spondylitis have silent intestinal inflammation even when they do not have digestive symptoms. Changes in the normal gut bacteria can weaken the intestinal barrier, allowing microbial substances to come into contact with the immune system, which activates dendritic cells, increases interleukin 23 production, expands T helper 17 cells, and encourages inflammatory cells to move to the entheses, where they sustain inflammation and contribute to disease progression. This helps explain the connection between intestinal and spinal disease.
The hallmark lesion in ankylosing spondylitis is enthesitis, which is inflammation where a tendon or ligament attaches to bone. This is the earliest and most important site of disease activity. Mechanical stress plays a major role in triggering this process, because repeated loading of these attachment sites can cause tiny areas of tissue injury, release danger signals from damaged cells, and stimulate the production of inflammatory cytokines that attract immune cells and maintain chronic inflammation over time. This is where the disease begins. Commonly affected areas include the Achilles tendon at the heel, the plantar fascia on the bottom of the foot, and the ligaments around the sacroiliac joints. The costochondral junctions in the chest and the corners of the vertebral bodies are also frequently involved. These sites are exposed to constant mechanical forces during standing, walking, and spinal movement. Importantly, enthesitis usually appears before structural damage becomes visible on imaging studies, so patients may have pain and stiffness for years before new bone formation or spinal fusion develops. Early recognition is essential.
Ankylosing spondylitis causes both bone destruction and excessive new bone formation, making it different from many other inflammatory joint diseases. This is a defining feature. Inflammatory signals such as interleukin 17 and tumor necrosis factor alpha activate osteoclasts, the cells responsible for breaking down bone, leading to areas of bone erosion. At the same time, pathways that normally regulate bone growth become overactive, including interleukin 22, bone morphogenetic proteins, WNT signaling, reduced activity of DIC-COP-related protein 1, and activation of mesenchymal stem cells, which together stimulate abnormal new bone formation and eventually produce syndesmophytes that can fuse the spine. This process leads to spinal ankylosis. Understanding how ankylosing spondylitis develops has changed the way the disease is treated. This knowledge guides modern care. Current targeted treatments block key inflammatory pathways, including tumor necrosis factor, interleukin 17, and Janus kinase signaling, helping reduce symptoms and control ongoing inflammation. Research is now moving beyond inflammation alone, with new approaches that target endoplasmic reticulum aminopeptidases, human leukocyte antigen B27, and the gut microbiome, while biomarker-guided treatment and precision medicine aim to match therapy to each patient, reduce disease activity, and prevent irreversible structural damage before it becomes established. This remains an important goal.
In summary, ankylosing spondylitis develops through the combined effects of genetic susceptibility, immune dysregulation, and environmental factors, which drive persistent inflammation at the enthesis, where tendons and ligaments attach to bone, and gradually lead to bone erosion, abnormal new bone formation, and eventual fusion of the spine. Our understanding continues to improve. This deeper knowledge has led to targeted biological therapies and Janus kinase inhibitors that provide better disease control and improve patient outcomes. Current research is focused on earlier diagnosis and more personalized treatment with the aim of preventing permanent structural damage. Thank you for watching. If you found this presentation helpful, join the StudyNeuro community on YouTube, Facebook, Instagram, TikTok, and X for more high-yield neuroscience education and practical clinical insights. Please like the video, share it with others, and subscribe for future content.